Very good morning to all of you. I once again welcome you all to this current video lecture. As you know that in the last lecture we started and completed the second important spectroscopy technique for the elucidation of the molecular structure. Now in the current lecture we are going to start the next important spectroscopic technique for structure elucidation is Raman spectroscopy and its importance. So our today's point of discussion is Raman spectra and the molecular structure. Now this Raman spectroscopy is a very important spectroscopy as far as the structure determination of a molecule is concerned. Now this spectroscopy is important from our point of view because this spectroscopy was invented by one Indian scientist, Sir C.V. Raman invented this type of spectroscopy. It is very important for our also. Now as far as this spectroscopy is concerned, in this spectroscopy we have to consider one important phenomena of light, and that is called as the scattering of light. In a previous two spectroscopy we consider the absorption of a radiation, but in this spectroscopy one important difference is there. In this spectroscopy we have to take into consideration the scattering of electromagnetic radiations. So what happens, this spectroscopy has the importance due to its different aspects as compared to the previous spectroscopy. Now this spectroscopy consists of this type of the arrangements in which we have to take a suitable source for the radiation. This is a suitable source is there. When the radiations are to be generated from this source, they are passed from this type of the lenses. So this light is called as the polychromatic light, means it has the all possible direction of its propagation, but we want for our study a monochromatic light. That's why this polychromatic light can be passes from a given filter, and that's why we get here a monochromatic light. This monochromatic light again passes from this slide is there. After this light get incident upon the sample, the sample molecule is there whose structure we want to determine, so that sample is taken at this part. So this monochromatic light then incident upon this type of the sample which we take in this tube. After the interaction of this monochromatic light on this sample, two type of the phenomena can be takes place. Some of the light get transmitted from this sample. Some of the light get transmitted from this sample. At the same time, some of the light get scattered. Some of the light gets scattered. This scattered light can be analyzed by a suitable analyzer which must be at a right angle to the direction of this type of the incident light. So this is a simple arrangement, experimental arrangement for this Raman spectroscopy is there. Means we have to take a suitable source. That source gives a polychromatic light. That light again get converted into the monochromatic light by passing through a suitable filter. Then this light can be incident upon a sample. Then some of the incident light get transmitted while some of the light get scattered with a given frequency, and that scattered light can be analyzed by a suitable analyzer. So this is nothing but the scattering of light. This is a scattering of light is there. Now what happens when this incident light can be interact with a particular sample? It has the some given frequency nu I is there. This nu I is the frequency of this type of the incident light. Now if the frequency of this incident light is equal to that of the frequency of the scattered light, if both these frequencies are equal, that is, we can say that if the frequency of this type of if the frequency of incident light, that is the nu I, is exactly equal to that of the nu S. Means if the frequency of incident radiation is exactly equal to the frequency of the particular scattered radiation, then this is observed by the scientist Rayleigh, and this is called as Rayleigh scattering. This is called as Rayleigh scattering. So what is this Rayleigh scattering? When the frequency of incident radiation is exactly equal to that of the frequency of scattered radiation, and this is observed by the scientist Rayleigh, and that's why this is called as the Rayleigh scattering. And the frequency of line is called as the Rayleigh frequency, and the frequency of this line is called as the Rayleigh frequency. So this is a very simple concept called as the Rayleigh scattering. So what is the Rayleigh scattering? The scattering of the monochromatic light by the given sample gives some scattered light if the frequency of this incident radiation is exactly equal to the frequency of scattered radiation, then this is called as the Rayleigh scattering. And in spectrum, the frequency of such a line is called as the Rayleigh frequency. So this is a first important phenomena, Rayleigh scattering. Now what happens? The scientist Smekil in 1923, the scientist Smekil predicted theoretically, that whenever such a type of monochromatic radiations can be interact with a particular source, a particular sample, then what happens? Some of the scattered radiations may have another type of frequency as that of the incident radiation. So he proposed theoretically that some of the radiations may have other type of frequency than that of the incident radiation. Next to that, the Indian scientist Sir C.V. Raman in 1928, the Indian scientist Sir C.V. Raman found that whenever these type of the scattered radiations we can analyze, it has some such a type of the scattered radiations which have some another type of frequency as that of the incident radiation. So due to such a type of the another type of frequency which obtained along with such a type of the incident frequency, so we can say that two type of the frequencies are there. Some frequency may be some frequency, that is a frequency of this incident radiation, it may be greater than that of the frequency of the scattered radiation, or some of the frequency in which the frequency of this incident radiation may be less than that of the frequency of the scattered radiations. So we can say that whenever such a type of the monochromatic radiations can be incident upon a sample, then these type of the different frequencies may be obtained in a scattered light, and that's why this type of effect or this scattering is called as the Raman scattering. This scattering is called as the Raman scattering. This is called as the Raman scattering, and this effect is called as the Raman effect. This effect is called as the Raman effect. So what is by this Raman scattering? According to Sir C.V. Raman, whenever monochromatic radiations can be interact with a particular sample, it gives a scattered light. That scattered light may have some frequency that may be another type of frequency as that of the incident radiation. Some frequency may have lower frequency as that of the incident radiation, or some of the scattered frequency may be higher than that of the incident radiation. So such a type of scattering which is obtained is called as the Raman scattering, and this effect is called as the Raman effect. Now what happens? Due to such a type of the scattering, we can say that there are two type of the different phenomena can be takes place. One phenomena is called as the Rayleigh scattering. Due to Rayleigh scattering, we obtain one line in the spectra. On the other hand, due to such a type of the Raman scattering, we again obtain some of the lines in a spectra. So these are the three different type of the lines can be obtained in the spectra of a molecule by such a type of the scattering experiment. So we can say that when this type of the condition is there, when this mu I, this when we say that when such a type of the condition is there, when this mu I is greater than that of the nu s, means when the frequency of incident radiation is more than that of the scattered radiation, at that time what happens, this delta nu, that difference, that is the Raman shift, is equal to positive. When such a type of the first condition is there, when frequency of incident radiation is greater than that of the frequency of the scattered radiation, at that time this delta nu, which is nothing but the Raman shift, that Raman shift is found to be positive. When such a Raman shift found to be positive, that lines are called as the Stokes lines. That lines obtained in the spectra are called as the Stokes lines. On the other hand, when the second possibility is there, now what is the second possibility? When the frequency of this incident radiation is less than that of the frequency of the scattered radiation, at that time what happens? The Raman shift, that is the delta nu, is equal to negative, and at that time the lines obtained in a spectra are called as the anti-Stokes lines. So these are the three different type of two different type of lines are there: Stokes lines, anti-Stokes lines, and the third possibility is that when the frequency of incident radiation is equal to the frequency of scattered radiation. When frequency of incident radiation is equal to the frequency of scattered radiation, at that time what happens? This Raman shift delta nu is equal to 0, and at that time the line which is obtained is called as the Rayleigh line. The line obtained in the spectra is called as the Rayleigh line. This is a Rayleigh line is there. So we can say that whenever a monochromatic radiations can be interact with a particular sample, at that time the light get scattered from the given sample, some of the light get transmitted. The scattered light may have these are three type of the different conditions. The first condition is that the frequency of scattered radiation may be equal to that of the incident radiation. At that time this Raman shift value is equal to 0, and the lines obtained due to such a type of interaction is called as the Rayleigh line. The second important thing is that when the frequency of this incident radiation is greater than that of the scattered radiation, that is at that time this Raman shift is equal to positive, and the line obtained due to such a type of interaction is called as the Stokes line. On the other hand, if the frequency of this incident radiation is less than that of the scattered radiation, the Raman shift obtained is equal to negative, and the line obtained due to such a type of interaction is called as the anti-Stokes lines. So this is the important interaction of a particular monochromatic radiation with a particular sample, which we are going to study the structure of that particular molecule. So this is a simple interaction. Now as far as these lines are concerned, the intensity of the Stokes lines are more than that of the anti-Stokes lines. So this is a spectra regarding the particular type of the molecule. So in this lecture we consider here the third important spectroscopic technique of a molecule, that is called as the Raman spectra. In this Raman spectra we consider a simple its experimental schematic diagram is there. That is a what is the experimental or what is its instrumentation of this Raman spectra is there. So in this case we take a suitable source. From this source we can form monochromatic light, then that monochromatic light can be interact with a particular sample. After the interaction of this sample, two type of the lights we get here. Some of the light get transmitted, some get scattered. Our importance is to that of this scattered light. When the scattered light is to be generated, this can be analyzed with the suitable analyzer. Now the frequency of this analyzer, when the frequency of this analyzer we found here with the suitable analyzer, then what happens? If the frequency of this incident radiation is equal to the frequency of the scattered radiation, at that time that scattering is called as the Rayleigh scattering, and the frequency which is obtained in the spectra is called as the Rayleigh frequency. So this is the Rayleigh scattering is there. After that, what happens? In 1923, the scientist Stokes proposed theoretically that the scattered radiation may have some other type of the frequency as that of the incident radiation. Next to that, in 1928, Dr. C.V. Raman proposed that, or he found that the scattered radiation may have some other type of frequencies as that of the incident radiation, in which some radiations may have lower frequency as that of the incident radiation, or some of the scattered radiation may have higher frequency as that of the incident radiation, and this scattering is called as the Raman scattering, and this effect is called as the Raman effect. Now whenever this Raman effect is considered, the three different situations is there. When the frequency of incident radiation is greater than that of the scattered radiation, at that time this Raman shift delta nu is equal to positive, and the line obtained in a spectra due to such a type of shift is called as the Stokes lines. When the frequency of incident radiation is less than that of the scattered radiation, at that time Raman shift is equal to negative, and these lines obtained is called as the anti-Stokes lines. On the other hand, the third case is there, when the frequency of incident radiation is exactly equal to the frequency of scattered radiation. At that time this delta nu is equal to 0, and the line is called as the Rayleigh line. So we get these three type of lines in the spectra of a molecule when we consider this type of the Raman spectroscopy. Among these three lines, this Rayleigh line is obtained exactly at the center of the spectra, while on the either side Stokes and anti-Stokes lines is there. The Stokes lines having the high intensity as compared to this type of the anti-Stokes lines. So this is a simple introduction regarding the second spectroscopic technique called as the Raman spectra for elucidation of the molecular structure. The remaining point from this spectroscopy we will study in the next lecture. So with this I stop here. Thank you so much.